Hello students, today let's discuss about post-translational modification in eukaryotes. Introduction. Post-translational modifications refers to enzymatic or covalent modifications of proteins that takes place during or after the synthesis of proteins. In eukaryotes, proteins are synthesized in the ribosomes, which are then further modified to give rise to functional proteins. Such modification can occur at the amino acid side chains or at the C or N terminal. The amino acids in the polypeptide can also undergo modification by addition or modification of functional groups. Some of the modifications are phosphorylation, glycosylation, lipidation, protein cleavage, formation of disulfide, etc. Post-translational modifications are important for cell signaling apart from the formation of functionally active proteins. Let us understand various types of post-translational modifications. In the first one, we'll see about phosphorylation. Reversible protein phosphorylation principally on serine, threonine or tyrosine residues is one of the most important and well-studied post-translational modifications. Phosphorylation plays a critical role in regulation of many cellular processes, including cell cycle, growth, apoptosis, and signal transduction pathways. Next, we'll discuss about glycosylation. Protein glycosylation is acknowledged as one of the major post-translational modifications with significant effect on protein folding, conformation, distribution, stability, and activity. Glycosylation encompasses a diverse selection of sugar moiety addition to proteins that ranges from simple monosaccharide modifications of nuclear transcription factors to highly complex branched polysaccharide changes of cell surface receptors. Carbohydrates in the form of asparagine-linked, N-linked, or serine-threonine-linked, that is O-linked, oligosaccharides are major structural components of many cell surface and secreted proteins. In the next form of post-translational modification, we'll see about ubiquitination. Ubiquitin is an 8 kilodalton polypeptide consisting of 76 amino acids that is appended to the NH2 of lysine in target proteins via the C-terminal glycine of ubiquitin. Following an initial monoubiquitination event, the formation of a ubiquitin polymer may occur and polyubiquitinated proteins are then recognized by the 26S proteasome that catalyzes the degradation of the ubiquitinated protein and the recycling of ubiquitin. In the next form of post-translational modification, we have S-nitrosylation. Nitric oxide is produced by 3 isoforms of nitric oxide synthase, also referred to as NOS, and is a chemical messenger that reacts with free cysteine residues to form S-nitrothiols. S-nitrosylation is a critical post-translational modification used by cells to stabilize proteins, regulate gene expression, and provide NO donors. And the generation, localization, activation, and catabolism of SNOs are tightly regulated. S-nitrosylation is a reversible reaction, and SNOs have a short half-life in the cytoplasm because of the host of reducing enzymes including glutathione and thioredoxin that denitrosylates proteins. Therefore, SNOs are often stored in membrane, vesicles, and interstitial spaces, and lipophilic proteins folds to protect them from denitrosylation. As for example, caspases which mediate apoptosis are stored in the mitochondrial intermembrane space as SNOs. In response to extra or intracellular cues, the caspases are released into the cytoplasm, and the highly reducing environment rapidly denitrosylates the proteins, resulting in caspase activation and induction of apoptosis. As nitrosylation is not a random event and only specific cysteine residues are S-nitrosylated, because proteins may contain multiple cysteines, and due to the labile nature of SNOs, S-nitrosylated cysteines can be difficult to detect and distinguish from non-S-nitrosylated amino acids. Next, we'll look into methylation. The transfer of one carbon methyl group to nitrogen or oxygen to amino acid side chains increases the hydrophobicity of the protein and can neutralize a negative amino acid charge when bound to a carboxylic acid. Methylation is mediated by methyltransferases, and S-adenosyl methionine is the primary methyl group donor. Methylation occurs so often that SAM has been suggested to be the most used substrate in enzymatic reactions after ATP. Additionally, while N-methylation is irreversible, O-methylation is potentially reversible. Methylation is a well-known mechanism of epigenetic regulation, as histone methylation and demethylation influences the availability of DNA for transcription. Amino acid residues can be conjugated to a single methyl group or multiple methyl groups to increase the effect of modification. N-acetylation. N-acetylation, or the transfer of an acetyl group to nitrogen, occurs in almost all eukaryotic proteins through both irreversible and reversible mechanisms. N-terminal acetylation requires the cleavage of N-terminal methionine by methionine aminopeptidase, also referred to as MAP, before replacing the amino acid with an acetyl group from acetyl coenzyme A by N-acetyl transferase, also referred to as NAT enzymes. This type of acetylation is co-translational in that N-terminase is acetylated on the growing polypeptide chain that are still attached to the ribosomes. While 80 to 90% of eukaryotic proteins are acetylated in this manner, the exact biological significance is still unclear. Acetylation at the NH2 terminal of lysine on histone N-terminal is a common method of regulating gene transcription. Histone acetylation is a reversible event and reduces chromosomal condensation to promote transcription, and the acetylation of these lysine residues is regulated by transcription factors that contains histone acetyltransferase, also referred to as HAT. While transcription factors with HAT activity act as transcription coactivators, histone deacetylase, also referred to as HDAC, enzymes are co-repressors that reverse the effect of acetylation by reducing the level of lysine acetylation and increasing chromosomal condensation. While acetylation was first detected in histones, cytoplasmic proteins have been reported to also be acetylated, and therefore acetylation seems to play a great role in cell biology than simply transcriptional regulation. Furthermore, crosstalk between acetylation and other post-translational modifications, including phosphorylation, ubiquitination, and methylation, can modify the biological function of the acetylated protein. Next, we'll discuss about lipidation. Lipidation is a method to target proteins to the membrane in organelles such as endoplasmic reticulum, Golgi apparatus, and mitochondria. vesicles like endosomes, lysosomes, and the plasma membrane. The four types of lipidations are C-terminal glycosyl phosphatidylinositol anchor, N-terminal myristylation, S-myristylation, and S-prenylation. Each type of modification gives proteins distinct membrane affinities, although all types of lipidation increases the hydrophobicity of a protein and thus its affinity for the membrane. The different types of lipidation are also not mutually exclusive in that two or more lipids can be attached to a given protein. N-terminal myristylation is a method to give proteins a hydrophobic handle for membrane localization. The myristoyl group is a 14-carbon saturated fatty acid, commonly referred to as C14, which gives the protein sufficient hydrophobicity and affinity for the membranes, but not enough to permanently anchor the protein in the membrane. N-myristylation can therefore act as conformational localization switch, in which conformational changes influences the availability of the handle for membrane attachment. Because of this conditional localization, signal proteins that selectively localize to membranes such as SRC family kinases are N-myristoylated. N-terminal myristylation is facilitated specifically by N-myristoyl transferase, also referred to as NMT, and uses myristoyl coenzyme A as a substrate to attach the myristoyl group to the N-terminal glycine. Because methionine is the N-terminal amino acid of all eukaryotic proteins, this post-translational modification requires methionine cleavage by the above-mentioned MFP prior to addition of the myristoyl group. This represents one example of multiple post-translational modifications on a single protein. S-palmitoylation adds a C16 palmitoyl group from palmitoyl coenzyme A to the thiolate side chain of cysteine residues via palmitoyl acyl transferases. Because of the longer hydrophobic group, this anchor can permanently anchor the protein to the membrane. This localization can be reversed, though, by thioesterases that break the link between the protein and the anchor. Thus, S-palmitoylation is used as an on or off switch to regulate membrane localization. S-palmitoylation is often used to strengthen other type of lipidation, such as myristylation or farnesylation. S-prenylation covalently adds a farnesyl C15 or general C20 group to specific cysteine residues within 5 amino acids from the C-terminus via farnesyl transferases, also referred to as FT, or general transferases, GGT1 and GGT2. Unlike S-palmitoylation, S-prenylation is hydrolytically stable. Approximately 2% of all proteins are prenylated, including all members of the RAS superfamily. This group of molecular switches is farnesylated, generylated, or a combination of both. Additionally, these proteins have specific 4 amino acid motifs at the C-terminus that determines the type of prenylation at single or dual cysteines. Prenylation occurs in the endoplasmic reticulum and is often part of a stepwise process of post-translational modifications that is followed by proteolytic cleavage by RCE1 and methylation by isoprenyl cysteine methyltransferase. We'll discuss about proteolysis. Peptide bonds are indefinitely stable under physiological conditions, and therefore cells require some mechanism to break these bonds. Proteases comprises a family of enzymes that cleaves the peptide bond of proteins and are critical in antigen processing, apoptosis, surface protein shedding, and cell signaling. The family of over 11,000 proteases varies in substrate specificity, mechanism of peptide cleavage, location in the cell, and length of activity. While the variation suggests a wide array of functionalities, proteases can generally be separated into groups based on the type of proteolysis. Degradative proteolysis is critical to remove unassembled proteins, subunits, and misfolded proteins, and to maintain protein concentration at homeostatic concentrations by reducing a given protein to the level of small peptides and single amino acids. Proteases also play a biosynthetic role in cell biology that includes cleaving signal peptides from nascent proteins and activating zymosins, which are inactive enzymes precursor that requires cleavage at specific sites for enzyme function. In this respect, proteases act as molecular switches to regulate enzyme activity. Proteolysis is a thermodynamically favorable and irreversible reaction. Therefore, protease activity is tightly regulated to avoid uncontrolled proteolysis through temporal and/or spatial control mechanisms, including regulation by cleavage in cis or trans and compartmentalization, as for example, proteosomes and lysozymes. Now let us discuss about importance of post-translational modifications. First, let us look into increase in proteomic diversity. Post-translational modifications are key mechanisms to increase proteomic diversity. While the genome comprises of about 20 to 25,000 genes, the proteome is estimated to encompass over 1 million proteins. Changes at the transcriptional and mRNA level increases the size of the transcriptome relative to the genome, and a myriad of different post-translational modifications exponentially increases the complexity of the proteome relative to both the transcriptome and the genome. Secondly, we have regulation of cellular activity. Additionally, the human proteome is dynamic and changes in response to a legion of stimuli, and post-translational modifications are commonly employed to regulate cellular activity. Post-translational modifications occurs at distinct amino acid side chains or peptide linkages and are most often mediated by enzymatic activity. Indeed, it is estimated that 5% of the proteome comprises enzymes that perform more than 200 types of post-translational modifications. These enzymes include kinases, phosphatases, transferases, and ligases, which add or remove functional groups, proteins, lipids, or sugars to or from amino acid side chains, and proteases, which cleaves peptide bonds to remove specific sequences or regulatory subunits. Many proteins can also modify themselves using autocatalytic domains such as autokinases and autoproteolytic domains. In another importance of post-translational modification, we have their importance in diagnosis. Consequently, the analysis of proteins and their post-translational modification is particularly important for the study of heart diseases, cancer, neurodegenerative diseases, and diabetes. The characterization of post-translational modifications, although challenging, provides invaluable insight into the cellular function underlying etiological processes. Technically, the main challenges in studying post-translationally modified proteins are the development of specific detection and purification methods. Fortunately, these technical obstacles are being overcome with a variety of new and refined proteomic technologies. Conclusion: post-translational modifications of protein is an indispensable step towards formation of a functionally active protein. It is also an important process in cell signaling which keeps the cellular machinery intact. Moreover, apart from the post-transcriptional modifications that can result in formation of different polypeptides through alternate splicing, the post-translational modifications also is responsible for the diversity of protein in a cell.